Excellent, very good. Good. So, hello everyone. I'm Asa Calow. I'm the second Mad Lab person on the bill this evening. As described in the introduction, I'm a professional technologist. I'm an amateur biologist, and as to my professional statement as a biotechnologist, that's heretofore unknown. So I'm here to talk to you a little bit about biohacking and DIY bio, and about what Mad Lab's involvement in this incredible movement. So, to begin with, I'm going to give you a brief and outrageously biased introduction to biology and biotechnology. So, biology, cover your ears, Stephen. So biology is particularly interesting as a science in that one of the original people who made the single most significant discovery in the science's history is still alive today, which is something that can't be said about sort of any of the hard sciences. So in 1962, it's very well known stories, and it's Francis and Crick, along with Maurice Wilkins and Rosalind Park, discovered the structure of DNA, so the genetic code to life. And that was 1962 that this incredible discovery was made. It took another couple of years before anybody really kind of actually sort of understood that this was quite what this was. It turns out that on the day that they discovered it, they made this model and showed this model. It was like, this is it, it's amazing, it's DNA. Meanwhile, elsewhere in the building, someone was going like, no, but wait, this guy's got this incredible thing just over here called a slinky spring. You've got to go check this out. So everyone else was down the other end of the corridor looking at slinky springs rather than looking at DNA. So biology, incredibly interesting, and since 1962, there's been this sort of incredible kind of sort of movement sort of all the way through. So a lot of the kind of significant kind of developments have happened in the sort of like 70s, 80s, 90s, and then sort of moving through. But sort of even like sort of the biology that was discovered in the sort of the 80s and 90s that have came out of DNA, our practices haven't moved on enormously from there in many respects, in that sort of a lot of the same kind of sort of processes still happen. But we've built up this kind of this incredible sort of multi-billion pound sort of industry, this kind of biotechnology industry, sort of like so building kind of like products and sort of applying sort of biological research to build up the industry. And so sort of biotechnology industry, we're reasonably familiar with sort of with the product, so incredibly important in, say, for instance, healthcare, sort of producing drugs, in fuel, in food, and the kinds of sort of companies that are involved in this that we know about they're multinationals. They're AstraZeneca, they're Pfizer, they're Monsanto. They're these enormous companies that that everybody's heard of. They're absolutely massive, and this is partly borne out by the fact that because it's such a new science, the amount of effort, the amount of research that's had to go into actually making sort of capturing the power of biology is incredibly large. There's a lot of money gets spent on research, and traditionally, sort of the biotechnology industry has been sort of worked in very much a kind of a sort of a binary type fashion. In the so like you hear about blockbuster drugs and like blockbuster drug development, and that's something whereby you have to plow an incredible amount of money into this complex science to kind of get to this sort of end goal, and it really is like fly or die. You spend sort of like hundreds of millions, sort of billions of pounds on producing this thing, and either it goes through several trials and it works, or it goes through several trials and people go, it's not actually that great. Or someone else has got a better one, and then the whole thing kind of crumbles. So there's just like everything just kind of turns on a die. So it's a real kind of sort of incredible kind of sort of thing. So biology, very, very complex. However, for the next part of the story, we're going to go back in time a little bit further. So over the past sort of couple of decades, there's been a quiet revolution in biology. And certainly around, say, for instance, the field of synthetic biology, which we'll come to in a little bit. And So what happens, in order to understand kind of what synthetic biology is and what the sort of, like, kind of new sort of forms of biotechnology are coming out, you have to go right back to 1841. And of course, you have to come back to the Northwest. So in 1841, Joseph Whitworth, he of like Whitworth Hall and the Whitworth Art Gallery, had this amazing idea for building like machinery and technology. At the time, everyone was using screws and like bolts to put all their machinery together. And what happened was that if you built a machine and one of your bolts broke, then you had to go and track down the same exact specific bolt. And so he had this idea of, so it's like, wouldn't it be great if they were just standardized? If you could just have someone over there that was making bolts, and then we all went and we all built our machinery to fit that standard bolt, and that would be great. So we can just go out and just buy a bolt and put it together, and we can fix our machinery. And wouldn't that be fantastic? And this was a significant sort of moment in the Industrial Revolution, sort of industrial history. Now, jumping forward another few years, so back around a couple of decades ago, a guy called Drew Endy started at MIT, so the Massachusetts Institute of Technology over in Boston. And his background was civil engineering, but he was coming into biotechnology. And his very first idea was, well, wouldn't it be great if, like, rather than everybody building everything from first principles, we had this kind of, sort of like Whitworth had the right idea, so let's copy that. So wouldn't it be great if there was this kind of sort of standard registry of kind of genetic parts, of kind of biological kind of sort of bricks that we can create new kind of biological sort of like things out of. And that would be fantastic. If we can really build this up, then we can turn it into, like as with electronics and the standardization of, like resistors and transistors and all these kinds of things has led to this kind of real sort of like explosion in the electronics industry. Then maybe something like this might be, might do the same for biology. So he came out with a standard registry of biological parts. And they were like, okay, great. So how are we going to fill this standard registry? So somebody else at MIT came up with a bright idea of having a competition. It'd be, so how about we hold a competition, an International Genetically Engineered Machines competition, iGEM, and then off the back of this, everyone that comes into this competition, we start to build up this set of parts, and then in successive years, people can come in and they can pick up the parts from the previous year and start kind of modifying them, adding their own parts, and kind of building sort of different kind of things out of them, these sort of different kind of sort of like genetic circuits, if you will. And over time, it'll kind of build up, and we'll end up with this kind of this fantastic sort of body of kind of well characterized, well understood kind of genetic mechanisms that we can kind of clip together to make things that are more more sophisticated. So iGEM. So that's where iGEM came from. It's a competition. It's an international competition. It's now absolutely huge. So sort of examples of things that have come out of iGEM, out sort of building these kind of circuits. So in 2006, the team at University of Edinburgh, they hooked together a couple of pre-existing biological parts to build a arsenic detector for water. So the idea being that you drop this this organism, this modified genetic organism, into some water, and you kind of grow it, and it would turn red in the presence of arsenic. Or it wouldn't if there isn't any arsenic there. It's great. It's a really kind of an interesting thing, quite sophisticated. So then fast forwarding a couple of years, so to 2012, to last year, there's lots of, iGEM has grown and grown and grown. They now have a high school division in the US and Europe. And one of the teams there was a high school team from Mexico. So you're talking under 16s in Mexico who are like, okay, that's great. So there's that arsenic detector over there. What about if we could modify that and tweak it a bit to just look for heavy metals? And so they did. And so they created this sort of genetically modified construct for searching for heavy metals in water, for testing for heavy metals. And that was a team of under 16s with help, obviously, but in Mexico who kind of sort of produced this thing. So it's kind of, it's like we've come a long way. But we've got this kind of idea that, okay, so it's still not simple. There's a fantastic analogy of kind of, sort of a bit like aviation. We managed to get the plane in the air, but we're not really entirely sure quite how. But the things that we can start to do with this stuff are more sophisticated. So where does MadLab fit in? So about 3 years ago, we started at MadLab a DIY bio strand. And so what DIY bio is short for do-it-yourself biology. Some people also call it biohacking, which I think is a particularly fun use of the word hack. And so our DIY biology strand, what's it there for? So DIY bio is something that started now 4 or 5 years ago. It's a sort of global movement. And the aim of this particular movement is to democratize and open up access to the tools and techniques of modern biotechnology. So this is great. So being a technologist and having lots of technologists at MadLab, sort of this kind of idea that, so you can take this kind of sort of unfathomable, squishy world of biology and then kind of make it something that's maybe a little bit more familiar to someone like me as an engineer or like a software developer. I was like, oh, fantastic! This sounds amazing. Let's do this. This is great. And the whole kind of thing of knowledge sharing, which is very much sort of built into the kind of sort of the core ideas of DIY bio, again, sort of like chimes with the whole sort of open source technology and the kind of sort of that sort of like sort of knowledge sharing that Simon was talking about earlier on. So like, oh, this is fantastic. This is amazing. Like, brilliant. Let's get going. Let's start a DIY bio strand. Let's stick a flag in the ground and go, like, brilliant. We're now DIY biologists. So one of the very first things that happened was a chance meeting at the Science Fiction Book Club, of all places, with a woman called Naomi Jacobs, who was then at Manchester Metropolitan University. We were talking about sort of DIY bio and going like, yeah, brilliant. We're like, okay, great. Well, we can help out with that and we can collaborate on that, and that would be that'd be fantastic. And from our point of view, it's just, oh, that's fantastic. I don't know the first thing about biology, so I'm glad that someone who actually knows a bit more about this stuff can come in and kind of tell us about it. So that's absolutely fantastic. So we started this DIY bio project. So the very first thing that we did was I was like, okay, great. So when do we get to start genetically modifying E. coli? We're like, yeah, you probably want to work up to that. So he goes, oh, okay, good. So let's just put a pin in that. We'll come back to that. You're like, okay, good. But let's start, a little bit kind of close to home. So I started off with microbiology. So the very first thing that we did was we built a Manchester microbe map. And we went out around Manchester and we kind of swabbed bus stops and then plated them up on nutrient agar plates, and then kind of sort of let the sort of various sort of things on these bus stops just kind of let them grow, to kind of see if we could get a sort of a picture, a bacterial microbiological picture of what the centre of Manchester looked like. And this was a lot of fun. If nothing else, then kind of sort of wandering around sort of like bus stops, kind of sort of getting really like funny looks as you're kind of there sort of scraping off bits of like timetable. So like, it was fantastic. It was brilliant, really good project. It's a microbe map, brilliant. So like, okay, so what next? Can we do E. coli now? Like, no. Okay, brilliant. So so what should we do? It's like, well, this is a global movement, so let's go and have a look and see what some of the other people in this area are doing. So we organised the first UK DIY Bio Summit. So again, you put a flag on the ground and said, everyone come together, we're going to do this amazing thing, and get everyone together in one place and sort of do something. Go like, okay, what does the program look like? It's like, I don't know, what can you do? Okay, brilliant. So we're going to have these sort of different groups. So we had like Cathal Garvey over from Ireland, who's a sort of incredible kind of character who's built a full, officially sanctioned biotech lab in the basement of his mum's house. We brought over a team from GenSpace over in New York, which was the first community biotech lab. And for a more general introduction to DIY bio, Ellen has got a talk about biohacking that's up on the TED website, which I can heartily recommend. We brought together Mark Dossier from the brilliantly named Hackteria, and countless other kind of DIY biologists and sort of amateur biologists, and sort of all got them together in one place. And we're like, okay, brilliant. So let's see what happens. And what happened was great. So we had all kinds of things happening. So we were like people building microbial fuel cells, so bacteria-powered fuel cells to see if you can get kind of like yeast to like switch on a light, which it turns out is quite difficult. They're quite lazy, yeast. But then we're also doing things like, so hacking USB webcams, so like a 3-4 pound webcam, taking it apart, putting it back together again as a microscope, which turns out to be incredibly easy. You can do it at home with a piece of blue tack. It's great. So we're kind of doing this and then starting to look at kind of, sort of interesting kind of pond life and all the rest. And also we had Oliver Medvedik from GenSpace was like, well, I've got some bits I can bring over. So he was doing actual personal DNA analysis. So personal genetic analysis, taking swabs, starting to investigate your own DNA just next to the kitchen in MadLab, using kind of Tupperware and various kind of sort of homebrew equipment. But it's, like doing like, what I'd like to think of as, like proper science, sort of using this kind of sort of like really kind of homebrew equipment. And that was the summit. So that was about a couple of years ago. And since then, we've kind of been on this sort of, we made a name for ourselves as kind of sort of putting on this brilliant thing, and it kind of, went off on a sort of on a tangent from there. So, this was a bit of an eye-opener. We're still kind of like waiting to do the kind of go and do the synthetic biology and do all that sort of stuff, but just kind of opening our eyes to this whole kind of sort of world of things that you can do elsewhere in biology, so microbiology, and sort of some of the sort of interesting things that nature just takes care of on its own, without us having to kind of tinker with it. This is kind of wealth of sort of biology that's already out there. So we've been on a fantastic journey. So this, as Rachel alluded to, we were invited over to the US by the FBI. It was heartily entertaining. It's a complete different story in itself. It was very fun. We went to BioCurious, which is the second but biggest, the excellently named BioCurious. Good naming in DIY BioWorld. So there's the brilliant name BioCurious, which is the largest community biotech lab in the US. And they were recently in the news for starting a glowing plants project, which picked up lots of money on Kickstarter. And I was like talking to a couple of guys about this. And their plant modification program consists of a grow box, and sort of sat in the corner of the lab. It's heartily entertaining, just behind a video projector. It's a really, really peculiar space. We also back in February, we organised a Lab Easy in London, which is kind of a sort of a combination of sort of speakeasy and kind of biotech lab. Sort of being kind of sort of like nefarious, sort of shady kind of amateur biologists. We thought it was best to set up in the shadiest and most amateur manner possible. So we set up kind of behind a nondescript door in London with kind of passcode entry, and set up a biotech lab in there. And running kind of all kinds of workshops, and again, just kind of as you go through this stuff, you just kind of find out sort of more and more. So we're doing kind of all manner of things from fermentation, so looking at the kind of microbiology of brewing and like pickling and all these kinds of things, with this amazing character called Sandor Katz, who wrote the kind of the Bible of fermentation, a book called The Art of Fermentation, which is amazing. We took baby steps towards the sort of synthetic biology, the goal of doing sort of amateur genetic modification workshops, staying very much on the correct side of UK law, but still kind of getting your hands on this stuff and being able to do it. What else are we doing? We do all manner of things. Cocktails, starting to look at sort of like the genetics of taste and palate, because it turns out that different people have inherited different kind of characteristics to the kind of flavours that they like. Kind of all manner of things, and it's been a real kind of, it's been a lot of fun. It's been a real journey. But this, so far, it's been interesting and a really, really weird hobby. But if it was just a weird hobby, it wouldn't be that interesting. But one of the things that we found is actually that when you start looking at kind of democratizing access to biology and biotechnology and really becoming a part of this kind of DIY bio community and sort of getting behind the whole kind of like knowledge sharing kind of sort of movement, there's some like incredibly kind of powerful things and incredibly interesting things start to kind of sort of tumble out of it. So like one of the key things for biology is that sort of necessarily a lot of the equipment and a lot of the kind of sort of chemicals and reagents that you use for doing sort of biotech are incredibly expensive. And as an amateur, you can't afford to kit out your lab like Monsanto can kit out a lab. So we're like, okay, great. So you start looking for kind of alternative routes in. And so the DIY bio mailing list is, this amazing kind of sort of resource of people going like, oh yeah, so that piece of equipment that costs like vast sums of money, yeah, you can make your own. It's kind of easy. It's like, something like a gel electrophoresis box, which is, this very sort of a high-tech bit of equipment. It's like, you kind of don't need it. You can do it with a piece of Tupperware. So we're like, okay, good. So we'll use the Tupperware version. And then, and the same with kind of sort of chemicals of, sort of things that have got, sort of incredibly long names. And you go like, yeah, if you nip down to like local store and turn over a packet of laxative, you'll find that it's basically the exact same thing that's sold as kind of like lab grade. You could just use laxative instead, and you get some very funny looks at the pharmacist when you're going back for coming back for my 10th box of laxatives. Thanks for that. But these kinds of things, sort of like amazing sort of this kind of things that come out of it. So this thing just behind me is a good example. So this is a PCR machine. PCR is the polymerase chain reaction. It's another scientific sort of like process. It was discovered in the 1980s by a particularly wild, particularly crazy guy called Kerry Mullin for DNA analysis, well, for DNA amplification. So if you're looking for a particular like tiny bit of DNA in amongst like a whole jumble of loads of other stuff, like, I don't know, you've got, say, like a thumbprint or a hair or something from a crime scene, and you want to, like, you just want to take out just a little bit that might be able to, like, fingerprint or identify that person, then what you need to do is take that tiny little, like, needle in the haystack of all the other DNA and amplify it, so produce lots and lots of it. So there was this technique that came up in the 1980s called PCR that allowed you to kind of replicate and produce lots of, like, focus in on a particular bit of DNA and look at that in particular. So 1980s, it turns out the equipment that you need for this is not very complicated. It's a thermal cycler, so it's a glorified oven. It heats to 3 temperatures over and over again in a cycle. And every cycle through the temperatures, it doubles the quantity of DNA that you're looking for while it goes through, along with some other ingredients. So we're like, okay, great, we can build one of those. So we did. So we made one out of some bits from B&Q, half a computer case, a light bulb, and the Arduino, the all ever-present Arduino. We have some outside if you're interested in this particular thing. A good platform for building kind of open source, kind of like low-cost hardware. So we built this thing. So sum total £50 rather than £2.5 grand. It was very good. It comes with some of the a few of the kind of sort of features missing, like, say, for instance, some of the fundamental safety features. So you can't just kind of switch it on and walk away from your desk. You kind of have to sort of switch it on and then kind of like look at it and cross your fingers it doesn't start smoking. But it works, and that's the key thing. And the whole kind of, and this whole sort of process of like reducing the costs of all this kind of equipment and all these sort of things means that, well, people who previously didn't have access to this stuff before can now, start to consider what this, what doing this stuff themselves. So maybe perhaps not necessarily this version, but there are some very nice kind of like open source versions that are kind of like laser cut and built properly. That that you can buy for a fraction of the cost of the commercial thing, and that means, for instance, that say for instance schools that couldn't previously teach biotechnology because the cost was too great. Well, if a school can't teach kind of modern biological techniques, then it means that all of the pupils in those schools, they don't have access to this to the technology that then allows them to even like think about, sort of consider taking a career in that kind of direction. So it's like by opening up access to all this kind of stuff, then you can start sort of introduce it into places where it's not been before. And also, sort of this idea of like the binary startup. There's still, not talking about, we're a long way away from when sort of like the kind of like DIY bio approach can kind of produce like sophisticated kind of blockbuster drugs or like anti-cancer treatments. Sure, there's, shouldn't kid ourselves here. This is, we're way away from that. But it opens up the possibility that, sort of rather than having these kind of binary startups where you have to plow in a vast amounts of money and then only to see them kind of sort of crumble at the end or become successful, you can start to kind of sort of maybe sort of like distribute some of the risk. And you can also start to do it in, you see kind of like garage businesses, it's like forming and like small, like especially in the kind of what you're seeing in the US now is these like tiny businesses starting in like the back of people's houses where they can start to kind of think about this, and like Hull over in Ireland. And it turns out there's an absolutely fantastic blueprint for a small-scale biotech industry that's already out there. A guy called Robert Carson has wrote a fantastic PDF about. In this particular blueprint, there's a company that falls into this kind of remit just down the road. They make a cracking pale ale. So it would be kind of great if you could start to sort of see kind of like biotech, as well as microbreweries. You start to see kind of sort of like biotech industry, or sort of people kind of being able to sort of do kind of sort of sophisticated kind of biotech work, on this kind of smaller scale than we've seen thus far. So, Mad Lab, we've gone off in a completely different tangent. So, it's like we've always had this sort of idea that we're going to come back to this, this goal of sort of synthetic biology, kind of, sort of a genetic engineering that we can do ourselves and kind of like really get into. We still haven't got there yet, but we've had an absolutely amazing time along the way because we found out an awful lot about other kinds of biologies and different things that are going on, which has been actually possibly more rewarding than if we'd just gone straight there towards the goal. But what happens next? Where are we going with this stuff? So one of the things that we're doing on Friday is we're going to go look at space, which is a potential lab space that we can get set up as an officially sanctioned biotech lab. And one of the things we've been doing is kind of like looking at, and rightly so, in Europe and the UK, there are an awful lot of regulations and laws you have to meet in order to be able to do this kind of stuff. But we've been talking to those guys and kind of working out what we can do. And on Friday, we're going to go have a look at space and, perhaps watch this, keep an eye on Mad Lab. There's, fingers crossed, we're going to be in sort of a position where we can start to kind of think about actually building a lab, a community biotech lab in the UK, so that people can kind of come in and start to kind of do this stuff. There have been rumours that iGEM, the competition that I spoke about earlier, are going to start allowing DIY teams from next year. So you can expect a kind of a cool running style Mad Lab team turning up within, if they let us, within the next year or two. That would be absolutely fantastic. If nothing else, I'm sure we'll have the most nicely designed poster. But that would be absolutely fantastic. And also, we're doing more and more workshops as well, and kind of and trying to disseminate all the kind of like knowledge that we've gathered along the way, and continue to do kind of sort of interesting sort of workshops and kind of public events around the kind of things that we've sort of discovered about the sort of DIY biology along the way. So we're doing a workshop at the weekend around for a genetic modification workshop. So people can come into Mad Lab, and we're going to make some E. coli glow in the dark. Because what's better than glowing bacteria? So those are the kinds of things that we're doing. So that's about it. Thank you very much.